robots for exploratory chemistry, because I thought we had a little bit already about discovery and supramolecular stuff. So it's a little bit more on the philosophical level, I hope, with some science in it. And my surname, you've seen it, so many consonants. I'm quite impressed that Adrian managed to pronounce it very properly. But I put a QR code if you want to look me up, or even have to try and copy all the letters. So thanks for having me here. And I just started in Durham a few months ago. So most of the work I'll be presenting today is from just before I started, but there will be bits and pieces that are a little bit newer. So the way I think about science, and I hope we all agree here, is we typically start by planning what we will be doing. We think about it, we design experiments, then we execute it. So we do the science, we analyze the results, we look through the data, we try and see what we've done and what the stuff we've made does, and then we draw some conclusions. So in drug discovery world, we call the DMTA cycle, so the design, make, test, analyze, but really that's kind of how we do most of the stuff. And we've been talking today a lot about automation, and I guess there's one brief slide on why I think it's worth automating things. So it increases reproducibility. So I am a physical organic chemist at heart with my sort of hydrogen bonding titrations training, and I believe we need to have a fair, better data, more of it, and we have to be able to trust our data so that we can then build some models and understand the theory. Hopefully in a longer term, it will reduce the costs, helps with health and safety considerations. You can do things without human intervention, and it's dangerous for the humans to do so, for example. We can accelerate the research timescales and hopefully free up some time for us scientists to do the thinking rather than the doing. So how do we do that in chemistry? Well, we can mine the data, we can use LLMs, we can use design of experiments, all of the tools we have at our disposal to plan the science. When it comes to execution, we have plenty of things. We can do flow chemistry, we can do syringe type things, we can do batch type things like computers, ChemSpeeds, or whatever else. The analysis part, it really has been semi-automated so far. We don't really run our NMRs manually sweeping the frequencies and then trying to plot the spectra. It's kind of all already automated. And then there is all of that emerging area of Bayesian optimization and supervised or unsupervised machine learning when it comes to drawing conclusions from the big amounts of data we make. So just to kind of give you a brief introduction on not my work, but what kind of started that part of the automation field I've been working in. There is an example from the photocatalysis field from Andy Cooper's lab from 5 years ago now, where the robot performed what is important here, 668 experiments over a continuous 8-day experimental setup. And that was optimization. So they tried to get as much hydrogen production as possible, basically, over a very large parameter space of what is in the reaction mixture. But what I'm interested in now is less so of the optimization, but more of how do we find something new? How do we actually do the science? And how do we discover a little bit more of an unexpected stuff? I think it was meant to be a slide here, but it's good I cut it out because we are a little bit short on time. So our response to that is that we want to have some autonomous synthesis workflows that do analysis and decision making. And by autonomous here, I mean with minimum human intervention. So in our case, we have a ChemSpeed in the middle here. So that's a liquid handling platform, heating, shaking, batch chemistry. So what you can do in a fume hood, you can do on a ChemSpeed typically. And what was important for us was to use equipment that we all have in the labs. So most of us will have an NMR somewhere or an LCMS, and we didn't want to have it all tied together so that the NMR is sitting idle for 24 hours while the reactions are stirring, because reactions are slow. NMR 5 minutes, reaction 24 hours. No point in having to make sure that that NMR can only be used for that workflow. So we wanted to use off-the-shelf equipment, expensive equipment, but readily available analytical equipment. And then we wanted that synthesis to be decoupled from that. So we used the mobile robots basically as fancy sample holders. They would move things around the entire lab, and I will show in a second how it was done. We wanted to have all of the results saved and published, including the failed reactions and the repeats and whatever else. And we wanted that to be scheduled from some sort of centralized host to make it a bit easier from data scheduling part. And for the interesting part, we wanted the analysis to be done a little bit in a way that human researcher would approach the problem. So you can see there is a mass spec type analysis, and then there is NMR type analysis, and they not always agree. So I think we had a question earlier today, how do you deal with false positives? And in my type of line of work, I want to encourage false positives, because if you just expect one possible product and you want the best possible yield of that, you get an interesting other reaction and you throw it down the drain because it doesn't conform to what you have already invented in your mind you're going to be doing. And that's an optimization type problem, not really a discovery type problem. So we wanted to have false positives, but we wanted to have false positives from orthogonal types of measurements. So basically to give them some creativity, but if enough of them agree that it's an interesting result, then it's probably an interesting result. If only one of them is a fluke, then that's also probably not really true. And then some have some sort of next step based on expert chemistry knowledge. So as you can see, the mass spec can have some hits and fails, and NMR can have hits and fails. And when they agree, that's a hit, and then it's being taken forward to the next step. So I said it's set in the lab. So that's the lab that I worked in before in Andy Cooper's group in Liverpool. So you can see there is the chem speed in the middle there, and a robot is coming up to pick up the samples for the LCMS. So does the typical robot dancing, moves away, puts them in a standard Waters Acuity UP LCMS. You can buy it off the shelf if you have enough spare cash. Then the robot goes away, another robot comes in from another side, takes a standard Bruker benchtop Fourier AT NMR rack of samples, puts them on the standard Bruker benchtop NMR arm for samples, and then the rest is carried on from there. If you really wanted to, you could come in the time when reactions are stirring and run your own spectrum on either of these instruments. So it's kind of free for you to use. So you need to apply it to a couple of hopefully interesting science problems. There is a little bit of supramolecular chemistry. I'll talk a bit later, but I thought the audience is a bit more real organic chemistry or real chemistry in principle. So let's talk a little bit about more drug discovery oriented approach. So you can see there's a set of simple reactions. I know urea coupling, that's not super exciting. There's some catalytic reactions. There's click chemistry or Sonogashira coupling. There's Nobel Prizes for carbon-carbon bond formations we learned earlier today. But the idea here is each of those steps can fail, and it can only really be taken forward if it didn't fail. Otherwise, there's no point in doing the reaction. And we wanted to reproduce the results at every stage. So, okay, one could argue this is not fully automated in a way, but I, a human, an expert chemist, would have to come in and tell the robot what is the successful reaction, what is not the successful reaction. But I only do it at the beginning. And once I've written it all up for this kind of small sub project, the set of rules that governed successes, like different from starting materials or the same as when we did it previously, just no change, or some sort of change or no change, are quite general. And we could very easily have puzzles that we could just put together for any new workflow that we're working on later on. So just to think about what decisions, what those puzzles would be, a trained human chemist could take. So for example, when run an LCMS, probably we want area percentage for this sort of high yielding step to be above some certain threshold, because when we have a forest of 10 peaks, we don't really want to be spending time on HPLC when it's an automated type workflow to try and separate it all. Maybe we want the NMR of the reagents to look different from an NMR of the reaction mixture. So reaction has happened, a very general, as you can see, prone to false positives binary classifier, but actually quite powerful when combined with the other criteria. For the scale-up step, okay, well, does it match what we did before? Is the LCMS the same? Are there the same peaks in the mass spec? Is the NMR the same? And when we are doing the diversification steps here, so we're coupling on larger bits and pieces, and I'm going to show you in a second what they are. They're like some HIV prodrugs or whatever. There are large molecules. That was an 80 megahertz magnet, so we kind of decided, okay, the NMR starts looking too messy for us to worry about. There are 3 solvents, very many carbons, protons, and no, let's just stick to LCMS and ask the software, is there a reasonable MZ value found in here? So Not all of the reactions worked, which is great because we wanted to, well, didn't really plan it before. We didn't actually test it before. We were a little bit too lazy for that. And one of them failed, and we could reproducibly fail it. So we were quite happy about it. So not everything was just super obvious. For the second part, you can see we've made a family of reasonably interesting products there with non-trivial structures. And actually, we even managed to find an unexpected product. So at compound 13 here on this slide, it has gone through all of the workflow being considered correct as correct synthesis. But actually, when we were writing it up and we'd run all of the high field spectra and repeated every reaction manually, we discovered, oh, something doesn't really work here. There is some cyclization going on, and there's a crystal structure, and it's a pretty nice, different, unexpected product. So we managed to actually get something unexpected, which we were quite happy with. But how do we actually expect the unexpected? And that was the problem of this combinatorial supramolecular host gas chemistry approach here. We wanted to not have a predefined criterion of it has to form an imine, or it has to make that coupling product, or it has to be this architecture. It has to be anything that's reasonable when it comes to being a metal organic structure. So not necessarily a cage, it could be a helicate, a polymer, or whatever else, but if it has stoichiometry that makes chemical sense, then it would probably be successful. So we had extremely loose criteria here. Basically, their starting materials had to look different than their products, and they had to satisfy basic chemical stoichiometries. And then we'd add some gas inside and see if they bind. So this is an example of the rules. I think I've run you through them already. So the reagents have to be different from the reaction, and then for the replication have to be identical, and for the host guest part, some peaks have to shift so that it's in interaction happening. And we've run it on this combinatorial mixture of compounds. And as I said before, we didn't really actually pick them. We picked one of them that we knew would work. Let's be honest, it's a published structure here. But there were also a couple of unpublished structures here that we didn't plan for because we just mixed things that we already had in the lab. And a single verification method, if you just rely on NMR or on LCMS, would sometimes give you a false positive result. So for example, I don't know, structuring top right, spectrum looks quite clean. I would probably actually work it up, but there's nothing in the mass spec. And that's probably because it was a little bit too labile as a metal organic complex to be stable under those conditions. Or structure on the top left, it's exactly identical to what we put in. So there is no reaction happening. So as I said earlier, we wanted to maximize the true discovery by minimizing false positives, but allowing those false positives for single methods to arise. And then that had to be tailored a little bit by expert chemists. So there is still some space for us as scientists to do things. I guess a nice comment I've once been given on this project was that we sort of reduce the middleman. So I guess we remove the postdocs. We have the students setting up a reaction and the big PI coming up with the idea, but the day-to-day of making more molecules and analyzing them, that we've taken out of the equation, which I guess helps the postdocs progress their careers faster and do other things. But yes, we've managed to identify a couple of new structures with one published cage and a helicate I haven't shown here because, again, you're not a supramolecular audience really. And then tried a battery of different possible guest molecules that can go inside. Mostly a set that showed some aliphatic aromatic signals so that we can demonstrate that it works in different parts of the chemical spectrum. Some that showed fast or slow exchange, which on an 80 megahertz instrument becomes a fun exercise because the timescales are very different from your 4, 5, or 600s. But yeah. So just to wrap up quickly, because I am aware that I am the last person between all of us and tapas, we tried to do an automated and autonomous chemical discovery platform, and encoding that decision making in chemistry is actually quite a hard problem, but we thought that it's easily transferable to a lot of different areas of chemistry, not supramolecular or not organic or not materials or not photocatalysis, but the governing rules are actually quite similar. So I hope I've managed to convince you that we didn't just have an automated workflow, but that there was some level of autonomy that was robots had, because it was the algorithms making decisions, not just us as humans later on, and that it wasn't just simply an optimization from very many different parameters, but we managed to actually explore different bits of space, not fully knowing what we were looking for. So not really obviously optimizing, because what is novelty, really. And I would like to thank all of the other people who were very heavily involved in that, and Andy and the funders, and you for listening to me. And if you have any questions, I'm happy to try and take them in 2 minutes.
Thank you very much, Philip. We have time for a quick question, I think.
Yeah, thank you very much. Very interesting. I have a question that's maybe more philosophical, but when setting out to do discovery, chemists usually trying to make something that fits an application need, not just make something. And so I wonder how you're going to tailor in future your discovery to fit a hypothesis of binding or some functional performance. Thank you.
So for me, the more interesting part of that is trying to understand where that function arises from. And I think having some physics-based modeling that gives us an idea of what structures we can target, and then having a more high-throughput data-based approach to collating the data and verifying those, I guess, what in the machine learning world would call input features, really, is a way of trying to bring some understanding into the function that for me is a more interesting sort of academic approach to a data-driven science. Because after all, we are here to discover something, not necessarily to make a slightly better yield for a reaction that already everyone else uses.
I think with that, I'm aware there are more questions, but in the interest of time, I think we should stop. So thank you again, Philip.
Thank you.